Hello, my name is Mr. Napoltz, and this is part 3 of our Central Dogma series of lessons. The Central Dogma is where we take DNA coding and we make it into protein. So how does that happen? In the last video, we went over the RNA processing and how RNA is cut down to its essential size, with all the introns containing non-essential info taken out of the mRNA. Now, the mRNA is in the cytoplasm, and now is free to find a ribosome to translate its coded message into a functioning protein. Now, remember, ribosomes can be found in the cytoplasm and on the endoplasmic reticulum. There are two different locations, just to clear that up. Ribosomes are also made up of a small subunit and a large subunit that are put together, kind of looking like a sandwich. It's made up of a combination of protein and another form of RNA that we haven't looked at before, ribosomal RNA, rRNA. That may seem a little strange, but just so you know, RNA is not just useful in carrying genetic code, it can also be useful for structure as well. So ribosomes are made up of protein and rRNA. The mRNA will be recognized and attached to the ribosome, and it will be put in between the small subunit and the large subunit, kind of like meat in a sandwich. But now it's here. And how in the world do you take this code and match it to specific amino acids? This goes back to how DNA code is read in the first place. DNA code is read in triplets, as in every 3 nitrogenous bases. So if we're going to look at this as, let's say, computer code, all right? Computer code has ones and zeros, but we don't read in ones and zeros. The computer has to translate it. It has to take these binary code of zeros and ones and turn it into language for us to be able to read. Even the letter A has to have a specific code of zeros and ones for the computer to display the letter A. So let's carry this idea into translation with ribosomes. If the ribosome is the computer in this picture, the codons, the triplets, are going to be the code for each individual letter. If a protein is the sentence, then the letters of that sentence are going to be the amino acids. But behind those letters, we have the coding for each individual letter, and those are the codons that you can find in the mRNA. As the process of translation occurs, we have another form of RNA that will recognize each of those codons. This is called transfer RNA. Transfer RNA is called that because it's going to carry with it an amino acid. A section of the tRNA, which will actually be at the bottom of its conformation, will have an anticodon, or a triplet of nucleotide bases that will match with a codon in the mRNA. And once it recognizes it, it will attach, and the amino acid that is already attached to that tRNA will be incorporated into the polypeptide. But let's go into more detail about how translation actually works. So we know how tRNA works, and we know that the ribosome is the site where this happens. But what is actually happening? There are going to be 3 steps. All right? There's going to be initiation, elongation, and termination, just like we had in transcription. The first thing to happen in this process is the small subunit binding to the mRNA. So this is before the ribosome subunits are even together. The small subunit individually binds to the mRNA and actually starts going down the mRNA sequence until it finds a start codon. Out of the 64 possible codons with 4 different subunits, there is only one start codon, which is an AUG codon. then the tRNA that is carrying methionine attaches to that start codon. The tRNA and the small subunit will be creating the initiation complex, where translation will finally begin. Then the large subunit will attach to this initiation complex, which officially begins translation. Step 2. Elongation. The large subunit is going to have 3 different areas in it, 3 different slots, as you will. One is the A site, then the P site, and then the E site. As we start things out, the first tRNA is going to be automatically in the P site. The A site, which is adjacent to it, will be where the next tRNA will match up with the codon in the mRNA. So let's imagine that a new tRNA has matched its anticodon with the codon in the mRNA and has brought along with it the amino acid that is supposed to be attached to it. As these 2 tRNAs are side by side in these 2 different sites, and the large subunit is going to interact with these 2 amino acids and will cause a bond to form between them. After the bond is formed, the tRNAs are going to shift. Not because the tRNAs themselves are moving, but rather because the ribosome is moving down the mRNA strand. And as they shift, the first tRNA is going to go to the E site, which is actually really just think of it as the exit site, because that is where that tRNA will be released out into the cytoplasm to get recharged with a new methionine. So the large subunit during this whole process is breaking the bond between the tRNA and the amino acid that it's holding when it's in the P site. And it's also forming a peptide bond between that amino acid and the new amino acid that arrives at the A site with the new tRNA. So as the tRNA leaves, the new tRNA that's having the new amino acid will shift into the P site, where now the A site is open for a new tRNA to bring its amino acid by matching its anticodon with the next codon in the mRNA. And this process will continue, adding on more and more and more amino acids, letting all the tRNAs go in this process until you get to a codon that actually stops the process. Step 3. Termination. Out of the 64 different combinations of codons you can have, 3 of them actually do not code for any amino acid. In fact, they only code for translation to stop. As the ribosome is waltzing down and reading the mRNA, once it comes to a stop codon, that stop codon is going to cause a reaction, which by and large just means that the polypeptide chain will be released from the ribosome and the tRNA that it was attached to in the P site. So you now have finished translation, and this whole complex of the ribosome and the tRNAs just dissipates. One final side note, you are not going to find many mRNA molecules that have only one ribosome translating them. Usually what you find, especially in our cells, is multiple ribosomes translating the same mRNA sequence. All right? So what it would look like is beads on a string. This complex is going to be called a polyribosome or polysome, where you have multiple ribosomes translating the same mRNA, and so they're going to be creating a lot of the same protein. All right, now we've gone through the general outline of how we go from DNA coding to mRNA, how that mRNA is processed, and then how it is translated into the functioning proteins that we see in our cells.